Good morning, good afternoon, everyone. Welcome to the MICD track as part of our MIDD 2023 conference. We are really excited about the session today. We are going to be working in the MICD track here as a track that is a new one to our MIDD conference, but where we see a really large focus of the use of PBPK and our machine learning tools as they help to support the regulatory experience as far as exposure assessments in the chemical industries. So we have quite a few speakers today that will continue to drive the need for modeling and simulation and the use cases there. Excited for that piece of it. Just a few housekeeping notes. We do take your privacy rights very seriously. By attending this event or participating in Q&A session, you are allowing us to contact you for follow-up. You may ask questions via the Q&A tab on your dashboard at any time. At the end of each one of these sessions, we will have some time for Q&A. And we will make sure that any unanswered questions, we do make sure we follow up to you in another session as well. So without further ado, I want to just thank Mike Bartels for kicking off our MICD track with the use of QSAR-based PBPK models for regulatory exposure assessments. Take it away, Mike, please.
Okay, thanks for inviting me to speak today. Today I'm going to share a few things with you primarily with the use of PBPK modeling for regulatory exposure, systemic exposure assessments for chemicals. Talk about modeling needs and timelines a little bit, how we've incorporated GastroPlus into some parameter estimations, and some specific case studies where we looked at the initial step of PBPK modeling, which is absorption estimates by both the dermal and dermal inhalation routes. This is a generic pipeline kind of thing you've probably seen many times. In the chemical industry, for well-tested molecules, products, there's a discovery development stage, registration, post-registration, re-registration. Throughout this timeline, there's all kinds of safety tests done, animal studies, ecotox, and things like that. In the animal space, as part of animal safety testing conducted where I was at Dow, and since then there's often early TK or pharmacokinetic modeling done just to interpret half-lives, partitioning, and things like that. Mid-phase, there's often some preliminary PBPK models developed to look at interspecies differences in uptake, systemic exposures, and things like that. Post-registration, as needed, there's often fit-for-purpose high-end PBPK models developed that support multiple species, including human, as well as multiple exposure routes. And then occasionally after that or beyond that, there is some need for some high throughput PBPK models that involve just quick analysis of some limited parameters like blood levels for a series of molecules to prioritize testing or to help support similarity in some PK parameters for some read-across assessments, especially in Europe. Throughout this approach, we've taken some steps to try and standardize some of these modeling approaches using some criteria on this page. I won't go over the details on these too much. We have them listed in a paper I'll cite on the next slide. But basically, we want to have modeling tools that are more standardized and fit for purpose models that support multiple exposure routes, have critical QSARs for absorption, metabolism, distribution, and things like that, support both PK and physiological based models, predict parent and metabolites, support species life stages, and then one advantage, if possible, is to find model tools that require a little coding and are more automated, and then also support batch modeling. In an evaluation of several tools, when I was at Dow, we selected GastroPlus as a tool for our modeling purposes. And then to evaluate the utility of this or support the utility of this, we did some evaluations mainly in the bottom here to support accuracy of predictions of FISCAM properties and some basic PK parameters like metabolic clearance and protein binding to help us understand the accuracy of the parameters that would go in to derivation of subsequent parameters and partitioning and all the components that, and outputs that come from the PBPK models. And one example of the output is we did an evaluation of some systemic exposure predictions against literature data. Now, as I mentioned, I'm going to go over a few slides here on some evaluations we have done and have been published in Zhang et al. 2018. The reference will be at the end. Here we showed for prediction of PKA values, the ADMET predictor module of GastroPlus showed good correlation between predictions and experimental data, and it was at least as good as other major QSAR tools such as Pipeline Pilot on the right. Similarly, with prediction of log p-values, lipophilicity had high correlation with experimental data and consistent with the accuracy seen for other common tools such as the EpiSuite program from the EPA. We've also looked at accuracy of predictions of metabolic clearance using empirical data obtained, donated by the EPA or shared by the EPA. And overall, we found that about 2/3 of the predicted metabolic clearance values that are CYP only based within GastroPlus were within 10-fold of experimental values. Same vein, accuracy of fraction of bound in plasma parameter was well predicted by GastroPlus with about 90% of the data compounds predicted within 30% of the empirical values. Shown on this slide, we have literature predictions of steady state plasma levels or blood levels for a series of chemicals and pesticides. On the two left columns in black font, data out of model predictions out of the literature from Wetmore et al., showing again predictions of steady state exposures, systemic exposures. And on the right two columns, we have predictions with GastroPlus using in blue font de novo predictions of metabolic clearance and fraction of bound in plasma, and then in purple using the same empirical parameters that were in the reference. In both cases, the values are fairly consistent between themselves and also with the second column, which are the data from the literature. So in essence, then, we feel that we have fairly good evaluation of GastroPlus for parameter prediction and systemic exposure predictions for a series of different chemistries. One of the other tools that we like to, one of the other phenomena that we like to be able to model also is understanding the nonlinearity in systemic exposures, or plasma concentration, tissue concentrations. One of the parameters that affects that is saturable oral absorption. A lot of our animal tox testing studies are done up to limit doses of 1,000 milligrams per kilogram. A recent manuscript in 2022 by an ILSI-HESI PBPK work group developed a generic simplified PBPK tool to illustrate saturable phenomenon like uptake, absorption, and elimination. In the absorption scenario, that generic model utilized a simple algebraic fit to data approach, shown in the blue and red lines here, where saturable absorption was fit based on a simple equation. That data, the terms for that data were fit to predictions with the GastroPlus compartmental GI model, which are shown in the black and brown dots here, that shows saturable absorption in the 10 to 100 milligram per kilogram range for these two chemicals. The parameters, again, from the fit to algebraic approach were fit to the GastroPlus model, showing the utility of an accurate, physiologically accurate model to as a stepping stone or initial phase of developing a more simplified open source models. Now, one of the simple case studies I want to go over is with tetrachlorobinefos, an animal health product. structure shown on the bottom here. Current risk assessments utilize derive human risk assessments based on dermal penetration studies conducted in rats. One of the issues with that kind of data is that it's fairly well known that rat skin is often more permeable than human skin to a lot of chemistries. And due to the fact that the rat skin is substantially thinner in all three primary layers of the skin, as shown in this, I think, excellent figure by Wee et al. In 2017. So to refine the risk assessment, get a better idea on dermal uptake in human directly, several things were done, summarized in this graph here. Overall, the species and dose dependent on dermal penetration for this molecule, TCVP, were either modeled in silico or measured in vitro dermal penetration assays or in vivo in the rat, in the red bars. A couple things to bring out on this case study that were quite useful. One is that the in vitro and in silico models and assays were quite consistent with the in vivo data in rat, both at the lower exposure levels as well as the mid and the high application levels. The trend was decreasing percent absorbed with increasing application levels consistent with a finite dose saturable dermal absorption. When we look at the absorption predicted and measured in human in vitro, we see trends that are consistent with the literature showing measured in vitro dermal penetrations in the blue bar substantially lower at all three exposure levels in human skin versus rat skin. And those same trends were observed in the GastroPlus models between rat and human dermal penetration, which are in vivo dermal scenarios, about three to fourfold lower in human consistently than rat. So these data are being submitted for publication soon, and hopefully will help refine the risk assessment for this material. The larger case study that I want to go over today is with work that's been done with a series of modified methylene diphenyl diisocyanate substances. These are materials that are utilized in the production of polyurethane and quite ubiquitous in manufacturing, and there's a lot of need to understand the risk assessment and systemic exposure sensitivity of these materials to support re-registration, in this case for the European geography. Under the read-across assessment framework developed by the EU, European Chemical Agency, an approach of grouping compounds based on analogs or categories has been supported and actually developed by the agency, that wherein registrants can group molecules based on similarities in properties, mammalian, ecotox, environmental, and utilize those groupings to fill data gaps in less well-tested molecules versus ones that are more well-tested. The advantages of this approach is it supports the use of predictive techniques in the field of chemical registrations, minimizes the use of animals in safety testing, which is an important thing, and in our case allows for the evaluation of individual constituents and mixtures. The chemistries that we're talking about in this exercise or case study are, again, 39 modified MDI substances. And this case study also has been recently published, and the reference will be at the end. But smallest molecules in the series are the monomeric forms of the methylene diphenyl diisocyanates, low molecular weight of 250. The middle molecules tend to be the multi-ring analogs of those, or the self-condensation products where we have heterocyclic rings in the middle of the dimeric or oligomeric species. And then on the bottom, we have prepolymers where any of the top molecules have been reacted with polyols polyol linkers to form prepolymers, which have molecular weight ranges that run up above 2,000. Common features of these chemistries are the fact that they're all quite lipophilic and contain at least 2 reactive isocyanate moieties. So again, the assessment that was done here for modeling is to look at the relative absorption of the 39 substances by the dermal and inhalation exposure routes, which are common routes in the occupational settings. The absorption is important in this scenario because beyond site of exposure toxicity, any toxicity that would occur after exposure would again be based on the magnitude of absorption. In the case of the dermal model, since these materials are reactive with isocyanate functionalities and are known to bind readily with things like glutathione and other proteins, we include pre-systemic metabolism in the epidermis and dermis layers of the skin. And we also utilize the solubility and vehicle-limited dermal uptake features built into the GastroPlus models. The inhalation model, a little more complex. It includes solubility and particle size, limited absorption into pulmonary tissues, as well as pre-systemic metabolism in the portal of entry tissues in of the lung. Mucociliary clearance, which is built into GastroPlus to account for clearance of unabsorbed materials, and then also pre-systemic chemical degradation in the GI tract. These molecules are, as I said, quite reactive and are known to undergo self-polymerization in the GI tract of animals where TK and tox studies have been conducted. And then after chemical degradation of a portion of the test material, solubility limited oral absorption is predicted for the unreacted parent compound. So as with our earlier generic evaluation of PK PhysChem parameters, we first looked at the accuracy of PhysChem parameter predictions for these series of molecules, because except for the monomeric small molecule forms, PhysChem properties of log P and water solubility have not been determined for these chemistries, again, because they have very, very low water solubility and they're reactive and will hydrolyze in water. So QSAR predictions were used to look at trends in lipophilicity and water solubility, plotted here against molecular weight, which is pretty consistent for both parameters. We see that the GastroPlus predictions within their ADMET predictor module give increasing trends of lipophilicity and decreasing water solubility with molecular weight, and similar trends are seen with a variety of other well-designed and validated QSAR tools. Due to computational limits within the GastroPlus program, we were required to use the GastroPlus water solubility because it only supports solubility limits down to 10 to the minus 10th, 8th mg per mil. But we were felt comfortable with that approach since the trend of decreasing water solubility was consistent between GastroPlus and 5 other modeling tools. So once we had parameters chosen for physical chemistry, those are utilized within GastroPlus to derive things like partitioning and dermal penetration and membrane penetration. We looked at some in vivo data sets to look at the accuracy of predictions for dermal absorption, looking at the lipophilic testosterone molecule with some in vivo data from the 1970s. We compared the solid bars of in vivo absorption data from Bartels et al., showing about 50 and 13% absorption of testosterone in rat and human versus the GastroPlus predictions, which are quite comparable. That showed that we had a good model to predict dermal absorption of lipophilic chemical. Moving over to one of our molecules then, where we do have some in vivo PK data, we looked at the dermal absorption of 4,4-MDI, which is one of the major monomer isomer. The solid bars again represent in vivo data. The blue is uptake into the skin. Solid green is absorption through the skin, and systemic bioavailability, either of parent compound or metabolite, since this was a radioactive study. Uptake into the skin was predicted to be consistent with the in vivo data, although GastroPlus only predicts absorption of parent, as well as bioavailability of only parent. The systemic absorption from material taken up into the skin was optimized utilizing the pre-systemic metabolic clearance parameter, and those resulting predictions would fit well with the in vivo data. I won't go into this in detail, but one of the advantages we like about GastroPlus is you can actually get a time course of uptake through the different skin layers, as well as predictions of total bioavailability plotted out in a scenario like this. These are some of the representative molecules from the lowest molecular weight up to some higher molecular weight materials. The full set of predictions of dermal uptake in blue bars and absorption through the skin in green bars are shown here. On the left, we have uptake and absorption predictions used for a test material applied to human skin using an acetone carrier solvent. And what we have here is for the monomeric species, the highest predicted absorption, uptake and absorption through the skin. The trend is to lower uptake and lower absorption with increasing molecular weight, dropping off to pretty much nil around the 700 to 800 molecular weight range. On the right, we had the same set of model predictions for the same test substances using a more lipophilic octanol carrier. Acetone is commonly used in lab experiments. These materials may often be formulated in more lipophilic solvents in occupational scenarios. So it's important to understand relative dermal uptake with a different carrier than acetone. In this case, the more lipophilic, less volatile carrier solvent has a predicted trend of similar relative uptake across the series, but overall lower magnitude, about tenfold. Now, switching to pulmonary absorption, again, we validated the model based on in vivo data from a rat study with radiolabel, where the material was determined based on several different data sets to be both inhaled and orally ingested via grooming post-exposure. About 90% was predicted to be, about 40% was predicted to be deposited in the lungs and 60% by direct oral intake. In the end, the absorption was determined by the authors of the in vivo study to be about 90% absorbed with about through the GI, with only about 10% absorbed through the pulmonary tissues. Optimizing our metabolic clearance and chemical degradation in the GI tract parameters, we were able to fit that data fairly well. Again, as shown we showed for the dermal penetration, the same kind of plot here for percent absorption of the parent compounds through the lung versus the GI tract, which represents, in this case, pure mucociliary clearance, is shown here for the 39 molecules. The blue bars are the pulmonary absorption is highest in the lung, decreasing to about the mid-range and dropping off from there. Mucociliary clearance is modest at the lower molecular weight range, increases a little bit in the middle range, and again dropping off above molecular weight 700 to 800. What's important here is that we've shown predictions of mucociliary clearance and oral ingestion of these test materials are possible by the inhalation route, which is helpful because there is certainly a data gap for these molecules on oral toxicity studies. Looking at the impact of the clearance parameters in the lung and GI tract was another thing we did, because the first model assumes consistent rates of clearance in either compartment across a series of molecules. if a molecule happened to have lower clearance in the lung and in GI tract, pulmonary uptake would not be predicted to be affected much, but uptake in the GI tract would be. Correspondingly, higher clearance in either compartment would give a modest decrease in pulmonary uptake, but a substantial decrease in oral uptake. Last thing we did in this exercise was to look at impact of particle size on predicted dermal uptake of these materials. And as you'd expect, as you get above the default of 2.5 micron, which is what our modeling was mostly done at, the larger particle sizes had lower, less deposition in the lung and lower overall pulmonary uptake. So in summary, we've shown the GastroPlus has been useful to provide predictions of phys-chem parameters and metabolic properties, as well as predictions of test substance absorption, oral dermal inhalation exposure studies. And this is especially important when we're talking about de novo modeling in the absence of measured parameters. Species differences in modeled absorption has been shown useful for trichlorvine-phos, and then dermal and inhalation absorption predictions across a series of MBI substances has provided data that we feel is useful for category-based read-across assessment that will be helpful to understand potential differences or similarities in systemic effects of these materials. List of references that I've gone over today are listed here. And then I'd like to acknowledge several groups of people who've worked on different components of this work. Several people at Dow Chemical, several people at Simulations Plus, the Hartz group that worked on the TCVP project, people at the III, or International Isocyanate Institute, and then a colleague at my firm. So with that, I'll stop. Well, I won't stop sharing, but I'll take questions.
Thank you so much, Mike, for a great presentation and a fantastic example of the way PBPK is being used across the chemical industries. Over the next few hours, really the thing that we want to continue to highlight is the use of PBPK and advent of machine learning tools, being different outcomes in the way that we think about some of the risk assessments in the chemical industry as compared to traditional uses in the pharma space. You mentioned some really good outcomes there, and there's some good questions in the chat that we won't be able to get to all of them, but most of them are really related upon how are we doing the translational outcomes with the way they're using animal models along with the in silico. From the outcomes that you presented on your previous few slides, is there a few with a minute or two left that you could highlight of where we need to continue to drive for more consistent usage of PBPK to help investigate particular questions or research challenges that people have?
You faded out a little there. I think you're asking if some of the recent data that I went over has some inconsistencies. Is that correct?
No, go to your 2 slides up, and I'll, the outcomes that you're presenting, your takeaway, your summaries.
Yeah.
Can you highlight where we need more adoption of utilizing?
Well, so one of the things that in dialogue with agencies, one of the things that we realize in the chemical industry space is that there's a preference for open source models that are completely transparent. All QSARs are well explained and defined, which is great for transparency. But the challenges that we find is that some of the proprietary QSARs, like those built into GastroPlus, are more accurate and more useful, especially in the cases of absorption, which is a parameter people don't think a lot about in the chemical space. Now, a lot of lipophilic chemistries, which there are a lot of, are absorption limited, especially at certain particle sizes by many routes, especially inhalation and oral. And understanding that impact on the design of animal safety studies is pretty important to know if the data that we're generating or we see is going to be relevant in human exposures. Because if we're doing exposures in animal studies above the point of any increased dose uptake between a mid and a high dose, there's no value in that top dose at all. So predicting that ahead of time helps us optimize our animal study designs and get the most data with the most appropriate dose levels for future risk assessments.